A very good morning, students. So here we are. So happy Sunday, anyways. So before we start the session, I would like to know if my technicalities, that is the audio and the video is clear for you so that we can continue with our class without any disturbance. All right. So students in YouTube, you can just put it in the chat message, okay, or the chat box saying that it's clear. If you find any technical difficulties, please let me know. Okay. So I would definitely expect a confirmation from the YouTube people. So please type yes or no if you have any technical difficulties with my voice or the video or the screen. All right. So let's get on with our class today and say where we left off. And then we try to build this understanding about what we have to understand from this chapter. Okay. So in yesterday's class, we understood about the Mendel's journey, the importance of understanding the terminologies that is related to this particular chapter. So those were very, very significant according to us, right? Or according to textbooks. So as we conducted the Mendelian monohybrid cross, we understood that Mendel, by monohybrid cross, what he did was mono is single, one, and hybrid is mixing a character, right? So he took one character and then he tried to, bring a hybrid among them. So the character he chose was the height of the plant that, as we know, the tall and the dwarf were the two characteristics are the two traits from a character called as a height. So here he took the pure tall plant and he crossed with the pure dwarf plant, and the resulting F1 generation was all of them were tall plants, but the genotype was capital T and small t. So when he crossed the capital T and small t with the capital T and small t in the F2 generation, he got 1:2:1 ratio. That is, three were tall plants and one were dwarf plant phenotypically, and genotypically we got capital T as one, capital T and small t as one, capital T small t and capital T small t as a genotypes, which were a couple of them, right? So after getting this, we understood that Mendel drew some of his observations, and he suggested that after seeing these ratios, that alleles appear in pairs. So this is very, very important. Law of paired factors. So always whenever we write alleles for a particular trait or a particular character, whether it is height, whether it is color, whether it is the shape, whether it is the position, anything you take. So we always represent them in pairs. For example, you go it like this, like capital R, capital T, okay, capital T, small t, capital R, small r. Are you understanding, students? So any allele that is represented, we always take them in pairs. So these paired factors, okay, are the representation of how the genes are present on a homologous chromosomes. Okay. So next one is law of dominance. What does law of dominance says? So we understood that in F1 generation, we got capital T and small t, right? So here, if you observe the alleles, both of them are dissimilar alleles, right? So the capital T is one type of allele and small t is another type of allele. But the expression of the F1 generation where all of them were tall plants, right? So here, if you look at what is happening, whenever alleles are in heterozygous condition, only one allele expresses over the other allele. The expressing allele, whichever the allele that gave rise to a character of this tallness, so this allele is called as an expressing allele. So this allele can be called as a dominant allele, and the one allele where its character is hidden or not expressed at all, So this allele is called as a recessive allele, right? So this is about law of dominance. So coming to the next point, uh, with respect to a monohybrid cross, is a very, very important statement that says Mendel's first law of genetics. So whenever we talk about Mendel's first law, we should not tell it's law of dominance, it's actually law of segregation. So law of segregation is also called as law of purity of gametes. So what is happening here? To understand law of segregation, we have to understand that how the cross went on, right? So initially, when Mendel took a tall plant and a dwarf plant, both of them were pure, correct? So when he crossed them in the F1 generation, he got capital T and small t. All of them were tall, but all of them were heterozygous. But when he self-pollinated this heterozygous plant, that is capital T and small t, when it was crossed with capital T and small t, so what we got was there was a dwarf plant in the next generation. In F2 generation, we did get a dwarf plant. So this shows that whenever the alleles are in heterozygous condition, so when capital T and small t was crossed with capital T and small t, when the gametes were formed, even the small t gametes were formed along with the capital T gametes. That is why it results in the dwarf plant, right? So if you observe, irrespective of the dominant character of the capital T, it didn't control the movement of the small t, correct? So in the next generations, even the dwarfness started appearing. So this is one proof that says whether it is a dominant trait or a recessive trait, it doesn't matter when the gametes are formed. So during gametogenesis, okay, when the alleles are in heterozygous condition, they segregate independently of each other. So this is a wonderful statement that Mendel derived out of a monohybrid cross. All right. So the next one is the test cross. So before we understand the test cross, students, okay, we have to understand why the test cross and what is the different types of crosses that we have. Okay. So first, initially, we'll start studying what are the possibility of a cross. What are the different possibilities of a cross? All right. Okay. Uh, all right. So now Mendel, when he tried to conduct a cross, right? So what happened to him? So the main reason for Mendel's success in genetic experimentation is his methodology. So we know that Mendel, when he chose pea plant, he had 7 contrasting characteristics that he was looking for. And these contrasting characteristics became a key for the success of Mendel's experiment, right? So if you look at all these characters, like the height of the plant, the color of the seed, the pod shape, the pod color, the position of the flower, etc. So for all of them, Mendel had pure pollen or pure gametes, we can call it as. How did Mendel was successful in isolating these gametes? Okay, so let us understand. So there are 2 types of crosses here. So one is called as the back cross. Okay, so what is a back cross? Back cross is, now I have a parent. My parents are, okay, my parents are capital T and capital T and small t and small t. So when they are crossed, they produce gametes called as capital T and small t, right? So I got F1 generation, and the F1 generation was capital T and small t, students. So once we have this, okay, so whichever the F1 generation or the children or the filial 1 generation, if it is crossed with a parent, it doesn't matter whether it is a dominant parent, whether it is a recessive parent, okay, it doesn't matter. You just simply cross an F1 generation with a parent. So that is called as a back cross. Okay, so that is the definition of a back cross. What is a back cross? You take an F1 plant and you cross it with a parent. What is a parent? Either it is a tall plant or it is a dwarf plant. So that is called as a back cross. Now, do we have any significance to the back cross? Partial beneficial. Okay, it's like partially helpful. It's not completely helpful. So that is why we split the back cross into 2 important cross. So that is, one is a test cross and other one is an out cross. Okay. So first we'll understand what is a test cross, and then we will understand what is a outcross. So now Mendel's idea of establishing what is the genotype of a particular plant, okay, had an experimental methodology. So that is called as a test cross. So how did he determine, oh, this is a heterozygous plant. So this is a homozygous tall plant. This is a heterozygous tall plant. How was he able to establish this proof? So that happened. So when Mendel, what happened is when he took a heterozygous, that is an F1 plant, okay, heterozygous plant, and he crossed with a homozygous dwarf plant. So what he got was his plants were the gametes were capital T type of gamete, small t type of gamete was crossed with here. There is only one type of gametes that could be formed. So what is the possibility, students? So we get the next generation capital T, small t, okay, sorry, capital T, small t, capital T, small t, small t, small t, small t, small t. So how did I get this? You already know. You just do this, right? You just cross like this and then you write it here. So if you see here, we got the tall plant and we also got the dwarf plant. So that means if the dwarfness appears when I do a test cross, that means the F1 generation or whatever the plant I crossed with the homozygous recessive plant is a heterozygous plant. Understood? So, and I'll just repeat here. So here I just took this plant. It was just a tall plant. Okay, I didn't know it was a heterozygous plant, and then I crossed it with a homozygous dwarf plant. If I do this cross, and in the next generation, if I find any dwarf plants, that is the proof that says that my plant that I crossed, the tall plant that I crossed, was a heterozygous plant. Understood? So next one. What if it was homozygous tall? So if you know it is homozygous tall, we already know what is a beginning of your monohybrid cross. So this is where we started the monohybrid cross, isn't it? So we know that all of them will be the tall plants in the F1 generation. So if we get all of them as a tall plant in the F1 generation, so that shows that it is a homozygous, okay, homozygous dominant plant or homozygous tall plant, correct? So Mendel actually did not directly take the heterozygous plant and crossed it with a recessive plant and established his results. He went on to do permutations and combination, and he ended up with this methodology. So what is a test cross? A test cross is conducted for what purpose? To understand the genotype of an individual plant or an individual organism. So if I want to understand the genotype of an organism, I take the F1 generation and I cross it with a, see here, I have an F1 plant and then I cross it with the homozygous dwarf plant. If I do it, I get the tall plants as well as the dwarf plants. So that shows that my F1 plant is what? It is a heterozygous one. Understood? Or my plant is a heterozygous plant. Understood? So this is called as a test cross. The cross made between a hybrid and a homozygous recessive plant is called as a test cross, and the advantage is to check the genotype of that plant. Understood? And the next one is outcross. What is an outcross? If you take an F1 individual, so you might ask, why not I take the homozygous dominant plant and then I cross it? See, that will result in outcross. So what I'm trying to say is, if I take my F1 generation, that is capital T and small t, and if I cross it with a homozygous dominant plant, so what do I get, students? I get capital T will go with capital T will go with another capital T will go with another small t, and capital T will go with another small t. So here, all of them I will get is a tall plant. So here, by seeing the phenotype of the plants, I would not be able to recognize whether it has a recessive gene, whether it is a homozygous or it is a heterozygous. So this type of a cross, where we have a F1 generation that is crossed with a dominant, okay, homozygous dominant plant. So that is called as what? Outcross. Okay, that is called as what? Outcross. So for us, which type of cross is very, very significant? Test cross is very, very significant. Understood? Students, are you able to hear me? Yes? Am I audible? Okay, are you able to understand? Please let me know. Okay, all right. So let's go to the next topic. So is Mendel's law of genetics, the first law of genetics, is applicable in all cases. So our thing is, we know that we have law of dominance and we have law of paired factors, and we know that there is something called as law of segregation. Law of segregation is universal and it follows everywhere, whereas this law of dominance is followed in all cases. So the first case we will take is the incomplete dominance. So these are certain cases where you don't have your law of dominance applying here. Okay, it does not follow the law of dominance. So Correns is a scientist who discovered this condition of incomplete dominance. So his experimentation on snapdragon flower or Mirabilis jalapa also shows the same thing. So what happens here is, when we have a red flower and when we cross it with a white flower, we do not get the red flower, but instead we get a white flower. So this is a very peculiar condition. So here, even in the Mirabilis jalapa, so again the same thing, the pink flower and the white flower will give you rise to the purple flower, right? So what is incomplete dominance? Now, as I told you, Correns is the one who discovered this incomplete dominance in Mirabilis jalapa. So what happens? Whenever we have two alleles that are present together in a heterozygous condition, neither of the alleles will completely dominate over each other. So that means, see here, students. So we have a condition where we have a red flower. We'll represent this red flower according to your textbook as capital R, and capital R is red flower. And then we cross it with small r and small r, which is a white flower, right? So this is a white flower. Now, when the gametes are found, we have capital R type of gametes and we have small r type of gametes, right? And then when the gametes fuse, we get the F1 generation, and the F1 generation where capital R and small r and all of them were pink, okay, which is very, very unique. Now, what we did was, in F2 generation, we took this capital R and small r and we crossed it with capital R and small r. So when we have this, what do we get here is capital R and capital R, capital R, small r, capital R, small r, and small r and small. If you see what type of plants we have here, we have red, we have pink, and we have pink, and we have white. Okay. So if you see the phenotypic ratio, it will be around 1:2:1. So we know that according to our monohybrid cross in the beginning, when we conducted, it was 3:1 as the phenotypic ratio. But in incomplete dominance, the phenotypic ratio deviates to 1:2:1. Mathematical deviations happen in rare cases, and this is one rare case. So in incomplete dominance, we get the both phenotypic ratio and genotypic ratio as 1:2:1. So what happened in incomplete dominance? Whenever the two alleles of, or whenever two alleles of different nature or heterozygous alleles were present, that is capital R and small r, whenever alleles are in heterozygous condition, neither of the alleles were completely dominant over each other. So that is called as incomplete dominance. So what is happening here is, the capital R is also expressing to certain extent, and small r is also expressing to certain extent. So both of them interact with each other and they will give rise to a new phenotype, okay, or a mixed phenotype. So this new phenotype, that is a pink color, that is called as, that condition is called as incomplete dominance. Remember this. Whenever in incomplete dominance, when we have two alleles interacting with each other, it will result in a new phenotype. So that is called as incomplete dominance. I'm specifically mentioning this statement for a particular reason, and that you will understand in the further class. Understood what is incomplete dominance? And another condition that we have is a codominant condition. Now, when it comes to codominant condition, before we understand the codominant condition, the example that we can understand the codominant condition is a human blood group. So in your class, if I ask you what are the possible human blood groups that we have, you will tell that it is A, B, AB, and O are our blood groups. That's what you would tell. Now, all of you who are telling A, B, and O, or some of you will tell A positive, A, B positive, AB positive, O positive, and all the negatives, right? So what are these A, B, and O? So these are the blood groups that are assigned to an individual based on the type of a blood antigen that is present on the surface of your red blood cells. So what happens is, when you look at your red blood cells, they are the cells and they have the plasma membrane. So the genes that are present in your cells, so they actually secrete a particular type of a glycoprotein, okay, or a polysaccharide, so that produces an antigen of type A or type B, or together A and B, or none of them, that is O. Okay, so there are almost 20 different types of antigens that are present on your blood group. One such important antigens is A type of an antigen, B type of an antigen, and then you have RH antigen that we look into, correct, right? That is rhesus factor that we talk about. So now, when we see this antigen, presence of an antigen, so this antigen is nothing but a particular polysaccharide that is secreted on the surface of your plasma membrane of red blood cells. So that is possible because of the presence of a particular gene. So now, having this antigen, okay, so that is when you will actually, I want A blood group only for the blood infusion, or AB blood group only is required, or O blood group is only required. That is what we tell when we go for blood donation. Now, why this is very, very required for us to understand? Now we have our medical reasons to understand this condition. One, we already know that we cannot mix the blood groups. That is, A person cannot accept the B blood group, nor the B blood person cannot accept the A blood group. The reason being, here, the antigens that are present on your red blood cells, they interact with the antibodies, and a particular type of reaction that causes in your blood called as deglutination reaction happens, okay, where the blood cells clump with each other, okay, and then the severe blocks and clogging of the blood happens throughout your blood vessels. So this might lead to minor complications to major complications, depends on how much of a wrong blood is infused into a person. Understood? So now, how did this antigen even originate? Now, if you look at the gene that is present, okay, on the chromosome 9. So on the chromosome 9, we have the gene called as 9q34 is a location, okay, where we have an I gene, okay, we have allele I and small I are the two types of alleles that are present on your, what do you tell, 9q34 spot of chromosome 9. So here, these alleles are responsible for giving rise to a particular antigen. So let us look into what are the different types. So for A blood group, we have IA, and then for B, we have IB, and then for O blood group, we have small I. So IA and IB are dominant alleles, and small I is a recessive allele. Understood? So based on the permutations and combinations of these alleles, we get a specific type of blood group. We'll look into it. Now, before we understand the genotype of an individual, let us understand the blood groups here. Now, if a person has a blood group A, that means he has A antigen on his blood surface, okay? So he has A antigen on his red blood cells, right? But what is there in his plasma? We know that our blood can be divided into two important portions. One is a cellular portion, other one is a plasma portion, right? So the cellular portion, that is the cell on the surface of the red blood cell, we have the A antigen, whereas the plasma will have anti-B. You look at here, anti-B, antibody it is, not antigen. So this antibody, okay, is the one that fights with the B blood group, okay, recognizing this B antigen when a wrong blood group comes in. Now you understand why the B blood group cannot be accepted by the A blood group, right? Similarly, the B blood group will have antigen B on its red blood cell, whereas the plasma will have anti-A antibody, whereas the AB blood group will have none of the antibodies in its plasma. That is why AB blood group, when there are no antibodies, antibodies are the ones that go and fight against the antigens. So that is why AB blood group people are called as universal acceptors. So they can accept any type of a blood group, right? So whereas when it comes to O blood group, you can see that they have both anti-A and anti-B antibodies in their plasma, whereas they don't have any antigens on their blood surface. So that is why the O blood group people are called as universal donors, where they can donate for A, they can donate the blood to B as well as AB, whereas they cannot accept any blood from any of the blood groups or any individuals having different blood groups. Understood? So these are the possibilities. Now, coming to how the blood groups happen. So let us talk about the alleles that are responsible for the A, B, and AB blood group. So for A blood group, an individual should have IA alleles or IA and small I, whereas the B blood group should have IB, or they should have IB small I, whereas AB blood group, the possible alleles will always be IA, IB. Got it? So the O blood group will always should be small I and small I. So if these are the genotype of an individual, then they will have, see, if the individual have IA, they'll have A blood group, IAI, they will have A blood group. Similarly, it goes to the B. For AB, it should be IAIB, and O, it should be INI. Got it, students? So now you have the possibilities of parent 1 and parent 2 and how their possible blood groups can be. I will solve a couple of them so that it will be easy for you to understand. So you take it as an assignment and you take this slide and then try to solve all the possible blood phenotypes, okay, using the genotypes, okay. Let's see. So they say that parent A and parent B are A blood group and the possible blood groups are A and O. So let's see. So we have A blood group. I say IA, small I. When they marry, IA and small I. So I get the offsprings IA, right? IA, small I. Then I have IA, small I. And then I have small I and small I. Here, wherever the A is present, since IA is a dominant one, all of them will have the A blood group. Whereas in one condition, I get the O blood group. Correct? So this is very curious, right? So let's solve one more. So I have one more condition. A parent A is A and the parent B is B. So what are the possible alleles? So I have here. So the parent A is A and the parent B is B. Now let's see here. IA, small I. So when it goes with IB, small I. Okay, so let's say these are the possible genotypes of the individual. So let's see. IA will go with IB, correct? Next, IA will go with small I. Then IB will go with small I. And then small I will go with small I. So you can see here, IA and IB will give rise to AB. Oh, sorry. Yeah, AB. Where is it? Yeah. Right. Why am I not getting the annotation? Yeah, here. All right. So I have AB blood group here. I have A blood group here. I have B blood group here. I have O blood group here. Bingo. So we have all the blood groups that are given here. So I have solved 2 students. So go and solve all of this. There is a specific reason I'm telling you this because all the time, in 90% of the cases, they always come with this question. If a parent A or if a grandparent has this blood group and the grandchildren have this blood group, what are the possibilities of the parent blood group? Or if the parents have this blood group, if one parent have this blood group and one child has this blood group, what is the blood group of a mother? So these are the possible questions that are possible that can be posed in your NEET examination. So how we can solve is we just look at the blood groups that are gone. See, we just looked at it and we established what kind of a genotype they might have. You understand my point? So using this technique, you can really establish the possible variants that we can get through these combinations. Okay, try to work this chart out. So this is very, very helpful. Okay, take this as a small homework. And as you go today to hostel, so take this as an assignment, at least solve 5 of them. Okay, it will be quite interesting. Okay, so let's go next. Next one is inheritance of 2 genes. So when it comes to inheritance of 2 genes, that means we are looking at the 2 characters, okay, or 2 different characteristics coming together. Okay, so coming to inheritance of 2 genes, we establish a law called as law of independent assortment. So before we go to the law of independent assortment, let us understand what is inheritance of 2 genes. For inheritance of 2 gene, Mendel actually took 2 characteristics together. So the characteristics that he chose for the dihybrid cross, okay, or inheritance of 2 genes, di means 2, hybrid means mixing of 2 characteristics, and he conducts a cross. So first one is seed color. seed color, and the next one is seed shape. Okay. When it comes to seed color, he has two possibilities. One is yellow, which is a dominant one. So capital, I'll write it as yellow. Other one is green. Okay, yellow and green. And seed shape is round and wrinkle. Correct? So round and wrinkle. Last time Dow students told that, sir, change your ink. I hope the reading is visible for you people. Okay. So if it's not visible, just be free to put a chat. Okay. Just let me know if there is any problem. Anyways, so let's go back here. So the seed color he chose was yellow and green, and the seed shape was round and yellow. Now, or sorry, the seed shape is round and wrinkled. So if you look at the dominant ones and recessive one, let us group them together. So I will write it as D for dominant and R for recessive. So here dominant one is yellow, so I'll write it as capital Y and capital Y. And the dominant shape is round, so I'll write it as small capital R and capital R. Whereas the recessive color is green, so I'll write it as small y and small y. Whereas the shape is wrinkle, so I'll write it as small r and small r. Remember, students, once you take the dominant letters, that is Y for yellow and R for round, so you do not write small g and small w. Okay, that is wrong. We have to use the same alphabets, and these alphabets, okay, have to be in small letters. I think you know the rules already. If you don't know, it's a very simple rule, but many of you, while writing your board examinations, you make the mistake of writing this Punnett square. Okay, so you just keep this in mind. So we grouped them together. What did we do? We took the dominant character to one side, and then we took the recessive character to one side. So in dominant, if we write it together, we can just annotate like this: round and yellow. Okay, and then we took it as wrinkled and green. So what is wrinkled? Small r, small r, small y, small y. Absolutely correct. Okay. So we have two characteristics. One is the shape, other one is the color. So we are bringing them together and we are conducting a cross. So let's see how our cross goes. Okay. So to conduct this cross, I have a few techniques, or it's very simple, which you already know. But anyways, you just observe this. It becomes so simple. Okay. So first, what do we have, students? So we call them as parents. So what do our parents have? We have capital R, capital R, capital Y, that is round and yellow, is crossed with wrinkled small r, small y, small y. Now, when you look at this condition, the parents, so they produce gametes. So imagine that I'm a parent. What is my ploidy, or what are the number of chromosomes that I have is diploid, isn't it? So when I produce gametes, so what type of gametes do I produce? I produce haploid gametes. That is, my gametes will have only n number of chromosomes, but my somatic cells or vegetative cells will have diploid number of chromosomes, isn't it? So similarly, we have represent parents as diploid condition. Okay, when we talk about gametes, so if I write G, gametes, so what are the possible gametes I can get? So you look here, out of R type of gamete, or out of Y type of gamete. So both of them will be together, correct? R and Y will be together, and then we cross it with small r, small y. Out of two, we take only one. So those are gametes. Now, when these gametes fuse, we get the F1 generation. So in F1 generation, capital R, capital Y, small y will be resulting. It just mix it up. So we get again round and yellow, correct? We get round and yellow. Now, again, Mendel wanted to discover what happened to this wrinkle and what happened to this green. Okay, he had an idea, but he wanted to establish even broader results out of it. So what happened here? So in F1 generation, what does he do? He self-pollinates. So why does he have to self-pollinate? He has to self-pollinate because he wanted to make sure what happened to this small r and small y. As simple as that. So in self-pollination, what do? We take capital R, capital Y, small y, and we cross it with capital R, capital Y, small y. Now, this is a very important and very easy step. All right. So I hope I can erase this. So let me erase this. Okay, done. Chalo. So this is very, very important step. So what do we have here? We are having an F1 generation that is capital R, capital Y, small y, and we are crossing with capital R, small r, capital Y, and small y, right? So while we're doing this, we have to again get the gametes. You remember, we have to get the gametes. Now, students, you just don't think too much. Just go with your instinct. What are the possible combinations I can make? Okay, so let us call this as a male plant. Let us call this as a female plant. All right. So what are the possibilities of male gamete? Okay, now repeat after me. I can get capital R and capital Y type of a gamete, colon, let us say, and I can get capital R, small y type of gamete, again, small r, capital Y type of gamete, again, small r, small y type of gamete. Correct. So these are polens. So again, here, can I get the same type, capital R, capital Y, capital R, small r, small capital Y, and then I have small r, small y. It's like a tongue twister. Okay. So now what do? I just write a Punnett square where I have, I just mentioned it as male or female. Okay. Now look here. They have just written in reverse order, but it means the same. See here, capital R, capital Y, capital R, small y, capital small r, capital Y, and small r, small y. Now, if you look at the above section, you see the same pattern, capital R, capital Y, okay, small r, capital R, small y, small r, capital Y, small r, small y. You see this? So you have to exactly write this in an exact order. So when you do this, your square becomes very, very easy. Now just mix and match these. Okay, capital R, capital R, capital Y, capital Y. Here, capital R, similarly, you just write the Punnett square. Okay, it's a long process, but try this Punnett square, very, very important one. So once we do this, if you observe what we have got, you will understand that you would have got around 9 of the seeds. Okay, we'll see here. Around 9 of your seeds were round and yellow. Around 3 of your seeds were wrinkled and yellow. Around 3 of the seeds were round and green. And 1 of the seeds were wrinkled and green. So this is a phenotypic ratio. So observing this phenotypic ratio, we can establish a very important law according to your dihybrid cross. That is nothing but law of independent assortment. So before we understand this law, why are we talking about this? If you look at these two conditions, that is wrinkled and yellow and round and green. If you see, wrinkled is a recessive character and yellow is a dominant character, right? The two different characters there. One is a shape, other one is a color, one is a recessive character, one is a dominant character of different nature. Now, when they are together, right? So they segregated independently, right? So it did not say, hey, I am a round shape. Okay, you are a green. You are not supposed to come and mix with me. It's not possible. So that didn't happen. So whenever, what does law of independent assortment says? Whenever alleles coding for two different characteristics are in heterozygous condition, they always segregate independently of each other during gametogenesis. So when the gametes form, it doesn't matter if you're a capital Y, it doesn't matter if you are a a small r. Okay, it doesn't matter. They independently segregate of each other. So that is called as a dihybrid cross law of independent assortment. Understood? So our phenotypic ratio would be 9:3 is to 3 is to 1, and our genotypic ratio would be 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. Okay, 1 to 1, 2 4 to 1 to 1 in simple terms. All right. So this is dihybrid cross, and this is what the conclusion of dihybrid cross. All right. So did you guys understand? Okay. So I'll just launch this quick poll so that I feel like little confident that whatever we are doing. Okay. Oh, sorry. I will end this. This is not the one I am supposed to launch. Okay. 1 second, students. Okay, I will just give you an A, B, C, D. So A for I understood. Okay, I'll write it as S here. Okay. Oh yeah, A for I understood. B for I didn't understand. Make it quick, no, please. Okay, the students who are in the YouTube, please, you can just put a chat message. Okay, just type in the chat box if you have any doubts, if you understood or if you didn't understand. All right. I think those students are here, right? And then we have Kutch. Okay, Kutch boys. So boys, please let me know if you are following me in the class. Whatever I'm saying is audible, or if you're understanding, let me know. Okay. So I can continue, right? Let's hope. Oh, I didn't launch. I'm sorry. My mistake. I didn't launch. Okay, here is your phone. A4 I understood, B4 I didn't understand. Okay. 15 seconds, students. Let's go faster. Okay, we can cover chromosomal theory of inheritance. Quite interesting. All right. Excuse me. Okay. So now we have to discuss about chromosomal theory of inheritance. So it's a very, very interesting topic to follow. Okay. So let's see what happens here. So when it comes to human beings, it's very easy to understand. You just have to look at around us. So, sir, when you just see our inheritance, you're just talking about tallness and dwarfness. When you look at ourselves, so we have so many characteristics that are not exactly matching our parents. We are quite unique. Okay, sometimes we are taller than our parents. Sometimes we are more smarter than our parents. Sometimes we are more darker than our parents. Maybe your mom has a better hair type than compared to you. Maybe your dad has a better hair type compared to you. Okay, you can see the characteristics and compare, and but you think that okay, I'm not quite matching here. Okay, my dad has a better characteristics than me. Okay, or mom has a better characteristics than me. Why am I so unique compared to my parents? So these are certain things that puzzles you. So that is when the chromosomal theory of inheritance comes to play to explain something very, very unique and very, very important. So basically, chromosomal theory of inheritance was proposed by Walter Susan and then Theodore Boveri. So now, when their work is basically on the movement of chromosomes during the meiosis. So we have studied what is meiosis and mitosis, and in meiosis we come across a very important term called as crossing over, where the recombinant nodule is formed and the recombination happens here. So his study was basically on how this movement of chromosomes influence the characteristics during the meiosis. Okay, so now what happened is they come to a conclusion that the chromosomes are not much different compared to that of genes. They are quite similar. They're very much similar, and they're very, very much parallel to that of the genes. But here what happens is the chromosome movement determines a lot compared to that of the genes movement. Okay, the gene is only one character, whereas a chromosome is something that carries hundreds of genes within it. Okay, so there might be a bulk transfer of information during this chromosomal movement. That is what they were trying to talk about. Okay, so now we know that the chromosomes and genes, all of them are present in pairs when it comes to diploid cells. And we know that homologous chromosomes are the ones that separate during the gamete formation. Now you know already what is homologous. That is 1, 1, 2, 2, 3, 3. So this 1, 1 will separate out during the gametogenesis. This one will go to one gamete and this one will go to the other gamete. That's how it happens. So when the fertilization happens, So the haploid gametes will come and fuse, and again the diploidy is restored in the zygote, right? So anyways, so we have certain colleagues that conducted the same experimentation. So one is Thomas Hunt Morgan and their colleagues. They performed this experimentation on chromosomal theory of inheritance. What are the variations that happens in the chromosomal theory of inheritance? Now Morgan's work is very, very significant. Morgan and his students, now what happened? They worked on Drosophila melanogaster, which is called as a Cinderella of genetics. It is also called as the fruit flies. Now, why Drosophila? Why did he specifically choose Drosophila? Now, as I told you before, the reason why we pick up a genetic organism is their size should be very, very small. They should be very simple to grow them in a synthetic medium in laboratory. They should have a very small life cycle, which they can complete within few weeks. So a Drosophila can complete within 2 weeks, and they show a large number of variations. Okay, so such as you can easily understand what are the wild types, what are the mutant types easily, and you can easily differentiate between what are the male and female. Okay, so and easily observable phenotypic characteristics are present in Drosophila, and the number of chromosomes are also very, very small. The size of chromosomes is only 8. So it's very simple to establish a study on how the Drosophila's genetics work. So that is where they specifically chose Drosophila. Understood? So now coming to this sex determine level, coming to this chromosomal genetics, understanding Drosophila gave us a better understanding that okay, chromosomes play a very, very important role in giving rise to a bulk amount of characteristics compared to that of genes. As I told you, genes are something that is singular. So it can only code for one character or a couple of characteristics. But when we talk about chromosome is something that controls a very huge amount of characteristics. It might be hundreds to thousands of characteristics that are bulkily transferred from the parents to the offsprings. Okay. So now having this idea of chromosomal inheritance, now sex determination is something that we can talk about when it comes to chromosomal inheritance. So observing these chromosomal patterns, Helkin in 1891, he traced a particular nuclear structure during the spermatogenesis of some insects. So when he observed this insect, 50% of our sperms received these structures, whereas another 50% did not receive these structures. Whichever received this structure, he named it as X body. Today, what we call it as an X chromosome. Okay, interesting, a story. Now, what happened? Using this movement of chromosomes or the type of chromosomes that are present, we can literally determine the sex. So now we have XX and XO type of sex determination. Now, in XX and XO type of sex determination, we know that the female has XX and the male have XO. So now what happened? The grasshopper has around 12 pairs of chromosome and 24 pairs. 12 pairs are there in total. 24 chromosomes are there, right? So here the total 24 chromosomes are only seen in females, okay, whereas the male will have only 23 chromosomes. Why is that? Out of 24 chromosomes, 22 chromosomes are called as autosomes. Autosome. So what are autosomes? Autosomes are something that codes for your somatic characteristics or vegetative characteristics. So for example, your liver, your heart, your kidney, okay, your intestines, your skin, okay, etc. So these are called as autosomes, which code for somatic characteristics or vegetative characteristics. And the rest one, that is the 23 and 24 chromosome in the female, they are called as what? Sex chromosomes. They are called as what? Sex chromosomes. And these sex chromosomes are very much responsible for formation of gonads, formation of reproductive structure, maturity, sexual behavior, etc. Understood? So now in females, there are two pairs. There is a pair of chromosomes. Those are called as sex chromosome, and those are nothing but XX. Okay, this XX is a sex chromosome. Whereas male has only one sex chromosome and the other chromosome is absent. O represents for there is nothing. So now what happens is, whenever you have, let us say that this is our male and this is the female, right? Whenever a male sperm carrying this X chromosome fuses with the egg, having this X body. So that will give rise to XX, right? Again, this will go with this and give us XS. So that will give us the females. Whereas when there is no chromosome, sperm, no chromosome means that doesn't mean that it doesn't have any chromosome, students. You have to remember that it does not have one sex chromosome. That's all. So when it goes with a female egg, which is carrying the X, which is having the X body, so that will give us XO and XO. So that is nothing but male. So remember, when it comes to XX and XO type of sex determination, it is grasshopper is an example for that. Understood? Okay. So the next one will go. Next one is XX and XY type of sex determination is very, very popular. So what is the example? So the determination happens in insects, for example, drosophila, and mammals like humans. Okay. So we know that here, we know that females are called as XX, male are called as XY. So here there is a term that I would like to give. So if you look at our XX and XY, the XX are produced by female. The female has XX and the male has XY, isn't it? When female produces eggs, so she will have 100% of X type of eggs, whereas when male produces sperms, 50% of the sperms will be of X type and 50% of the sperm will be of Y type. Since the male produces two different type of sperm, he will be called as male heterogamy. Male gamete means gametes. When different type of gametes is produced, that is called as male heterogamy, students. Understood? Now we already know we have 23 pairs of chromosomes. Out of 23 pairs of chromosomes, we have 44 autosomes. I told you what are autosomes already. And then we have XX as a sex chromosome or XY as the sex chromosome. Now, when an X egg meets with X sperm, so that will give us XX, that will give us girl child, correct? So whereas X egg goes with Y sperm, so that will give us XY, so that is giving us boy. Understood? So this is chromosomal basis of sex determination. This you remember, male heterogamy. Okay, next one. So you can see here the same thing is depicted in this picture too. So next one, ZZZW type of sex determination is quite interesting. So what is happening in ZZZW type of sex determination? The example for this is birds. So you can see here the male birds have homogamy, whereas the female bird will show heterogamy. So what is happening here? The male shows ZZ type of a sex chromosomes, whereas the female shows ZW type of sex chromosomes along with the autosomes. So when a male produces the sperms, all of the sperms you can see here will be of only Z type, whereas a female will produce two different types of eggs. That is one Z type of eggs and other one is W type of an eggs. Correct? So whenever we have a sperm that is going and mating with ZZ, so we have the females. Okay. Whenever we have our sperm going with and mating with the W type of an egg, then we have a male. So here, here it is male. Here it is female. Okay. Here it is female. Sorry, people. All right. So here, what we can understand is the male produces similar type of gametes, so it is called as male homogamy, whereas the female produces two different gametes. That is why it is called as female heterogamy. Understood? Okay. So actually, we had to talk about the sex determination in honeybee. Okay. So honeybee sex determination is number-based sex determination. So I'll just write it here. Okay, the slide is missing, so forgive me for that. Okay, again, I'm not drawing a straight line. Okay, almost. Okay, so now what do we have? We have a female honeybee, and then we have a male honeybee. Okay, so now what do we have here? The female has 32 chromosomes, whereas the male have 16 chromosomes. We call it as haplodiploidic sex determination based on chromosome numbers. So we know that the organism should be in diploid, the organism should be in the diploid state. Here, the male is haploid. Now, what happens is the female, when she produces the gametes, she produces through meiosis Okay, meiosis. And then when she produces meiosis, she produces eggs which are haploid. Whereas the male, when he produces the gametes, he goes under mitosis and then he produces the haploid sperms. Now, if the sperm meets an egg, they will fuse together to produce a zygote, which is 32. This will develop into a female. Interestingly, sometimes the sperm will not meet the egg at all. So this egg will undergo parthenogenesis. Okay, parthenogenesis. I'm not able to see. Anyways, parthenogenesis. Parthenogenesis is fertilization happens without the fusion of gametes, right? So the fertilization happens without the fusion. So the parthenogenesis happens and it results in a haploid male. So this is called as haplodiploidic sex determination or chromosome number based sex determination. All right, students. So we come to the conclusion of the session. So we have linkage, recombination, and then we have six disorders. Okay, the Mendelian disorders and chromosomal disorders. I'll definitely try to cover up in 15-20 minutes in the coming sessions. Okay, we'll steal the time from the other sessions. Very, very important because that is why I'm trying to go in detail with respect to genetics, as well as when we talk about molecular genetics. We have to talk in detail so that we understand the concepts very thoroughly. That will help us solve questions if we know concepts better for our need. All right. So thank you very much for attending the class. So I hope we become more interactive in the coming sessions. If you have any doubts, the chat box is open for you. If you don't have any doubts, have a nice day. Have a good Sunday. I'll see you in the next class. Thank you.